So I'm very, very happy to have with us today Artur, who will present us some very exciting work that they're doing at Causaly. I have been in discussions and being informed about Causaly since they were a startup. I think now they are, I don't think they call themselves anymore a startup because they're really doing great and they've done a lot of interesting things. And thank you, Artur, for accepting our invitation. I'm really very, very happy to have you with us today.
Thank you very much, George. My pleasure is all mine. Thanks for the invitation, and thanks to Elena also for organizing and supporting me to deliver this. I hope this will be very informative and helpful. And yeah, I'm happy to start. So yeah, once again, thank you, George. Indeed, we have met well, a few years back now, right? We were in our very early stages of building the company. We still call ourselves a startup. We just became a bigger startup. And I'm very happy to tell you about Causaly today. My plan is to go over several slides to tell you about the problem that we are working to solve, about our technology, about the team a bit. I'd like to start, of course. And then I think it would be very useful if we actually get to look on the product that we are building together and then see a few use cases. That's my plan. So yeah, and then as George mentioned, I'll be happy to address any questions at the end of the call. So let me start. I'd like to speak about the company a little bit. We were established in 2018, and we are relatively young. We are headquartered in London. However, I have to say half of the company today is based in Athens. And before pandemic and hopefully after pandemic, we were very frequent guests in Athens and miss it a lot. Our mission and vision in the company is to democratize scientific evidence and findings to a very large group of researchers in biomedicine and healthcare. And we want to do that without assumption that they have data science expertise. So how do we do this? We build a machine reading system, a platform that could extract evidence across tens of millions of documents from free text and represent them to our users. And that solves a really big problem today already for many. And we hope that in succeeding in this, we could really deliver a transformation how biomedical research and access to evidence, knowledge, discovery and acquisition are done. We are very proud already to work with very large companies, our partners, customers in the area. You can see a few of them, and it reflects right on the size of the problem. The size of the problem is, of course, large. It's, well, large would be probably quite an understatement. Over recent decades, our scientific output delivered us a rather exponential growth in the knowledge, right? In digital knowledge, especially. This applies across many science domains, but specifically for biomedicine, our understanding how biomedical processes work today resides in around 32 million academic publications and growing rapidly, right? By 100,000 documents each month. So how can people work with this amount of knowledge, amount of documents today? Well, it's there's only one way. It's a status quo, and what we have to do is we only can create various keyword searches, Boolean queries, and in the index databases, try to find what we're looking for. So today, if I wanted to, for example, search for biomarkers of lung adenocarcinoma, what I would do is I would go to a PubMed and I would input this search query, keyword search, lung adenocarcinoma and biomarker or receptor or antigen. I mean, of course, my query would be more robust and bigger, but for the sake of the example, this is what I would do very quickly. What I would get is around 17,000 documents. Check that number today. Yesterday, it could already be different. It could be already different today. Now that I have 17,000 titles basically paginated in front of me, the only thing that I could really do is I need to screen and filter these initial results. So this would take me days and weeks of reading time, possibly causing me reading fatigue. And I would, of course, have to work out some sort of a trade-off where I can either do more reading, I have to do more reading, and or sacrifice the sensitivity of the search if I cannot do reading anymore. And one thing that we very much believe is this way is inherently limiting for knowledge discovery and hypothesis generation. Because if you think about it, my Boolean query searches for something specific, and if some finding was not already encapsulated in my query, it's very unlikely that I could find it, discover this knowledge that I wasn't thinking about through interfacing this basically 17,000 of titles. Only unless, of course, I would read all of them. So the status quo basically today does exactly this. It's a keyword search, and what it does in essence is it finds documents. What do we do differently? We at Causaly find evidence first. What we build is a machine reading platform that is incredibly fast in reading text, and we already have pre-read sort of these 32 million documents, right? We extracted all the evidence that we could find, and we packaged it in the knowledge graph, about which I will speak a bit later. So the same question that I was showing in the previous slide, on Causaly, I would ask, semantically, I would ask, what are the biomarkers of lung adenocarcinoma? What I would first see is a slightly more than 3,000 various biomedical concepts, genes and proteins, enzymes, receptors, which are shown to be or studied to be as biomarkers or biomarker candidates for lung adenocarcinoma. And this snapshot of a dendrogram is actually the very first thing that I would see, right? So I would not be looking at rows of titles. I would actually immediately get access to a landscape of what is the evidence for biomarkers of lung adenocarcinoma across the entire publication knowledge base, in this case from PubMed. So evidence from abstracts and full text surfaced within seconds. That search would be executed. We get an instant bird's eye through very highly visual tabulated data. And if any of these findings, they interest me, then I could immediately go click on them and get the evidence from the articles themselves. Where did we extract this? The sentences, from which section of the article they're coming, and others. And this is not only our hypothesis, this is also something that we are witnessing. These are these wow moments, right? When people didn't realize something and saw some concept, and then they click on it and they say, oh, wow, I didn't actually realize there is this relationship or this connection. We don't get them on a daily basis. They are rarer than that. However, they do happen. And we believe very much in this power of knowledge discovery that our product can deliver. I mentioned the word evidence several times. I think it would be valuable to align on what we mean by evidence. I have a few examples here, but I would like you to think of first of our primary data unit, an academic paper, for the sake of the example. Academic papers, they are structured set of sentences which carry certain narrative, right? And they have an article title, they have abstract, they often normally have various sections, introduction, related work, results, findings, and so on. But what in essence we look for in them when we first scan them, we look for single evidential sentences, a few of them normally in a paper. And after we understand the major finding of this paper, we then decide, do we read it further or not? And this is what our platform does. It goes and the machine reads every single sentence from all these papers and extracts evidence that we could show first before people would have to go and let's say read 17,000 abstracts and then full texts. So let's look at two examples. Our machine reading platform can really perform semantic relationship extraction. I'll elaborate on it on the next slide. But let's look at the first two examples here for metformin. In the first one, author said metformin exerted an overall positive effect on the clinical and endocrine metabolic features of PCOS. And on our platform, you would see this relationship as metformin downregulates PCOS, which is polycystic ovary syndrome. And our system would go and see metformin as an organic chemical or drug, and it would recognize PCOS as a disease. And understanding linguistically and lexically the construct in this sentence, it would say that it is a downregulate relationship because a drug exerted an overall positive effect on endocrine metabolic features of a disease. If we change basically a few words, keeping the structure pretty much the same as in the second example, where author said metformin administration was accompanied by a higher incidence of abdominal discomfort, while the structure is the same, now we see that metformin administration results in higher incidence of a disease. And this we would classify as a side effect. So we don't basically count statistically and infer relationships from, for example, co-occurrence. We really do extract evidence semantically, and we do it by using our natural language processing pipeline. It's I think it approaches around 30 stages right now, but I outlined 5 of them, which are quite valuable to discuss here. And I have the example on the bottom left, which would also be good to sort of dissect for better understanding. So given a sentence on the bottom left, recent reports indicate that the chronic consumption of coffee leads to reduced risk of ALZ disease. The way to translate how our pipeline works on a high level, it would basically read the sentence and try to find a subject predicate object agreement between two entities. And we have one here. It's a coffee and ALZ disease, which is Alzheimer's disease abbreviation. And there is a predicate SPO agreement there. It says that coffee leads to basically reduced risk of ALZ disease. So we have the two entities, coffee, ALZ disease, and we have leads to. However, that's not all the information, right? Coffee leads to Alzheimer's disease would mean completely something else. So we also try to understand if there are events and what are the head sort of keywords on both sides of this agreement. And for coffee, the event is consumption of, which could only be expressed as an intake or increase of coffee. And for Alzheimer's disease, the event is reduced risk. So basically, increase of first entity leads to reduction of second entity is now way more informative for us and more comprehensive linguistically. And what we would do next is we would add the semantics. We would add the context. Coffee, in our case, is a substance, and ALZ disease is a disease. So what we end up with is there is a substance which leads to reduction of risk of a disease, and we would translate this as a down-regulate relationship. This evidence. We would do more with the sentence. Of course, we would look at the various clausal complements and the various hypothetical or negated cues, and we would use them all to evaluate the sentence, score this sentence linguistically. How strong is this relationship expressed by an author? Is it hypothetical? Is it negated, and others? And one more thing to add here is our solution is generalizes across all domains within biomedicine and surrounding areas, right? There are around 148 categories similar to disease, substance. There are therapeutic procedures, living beings, nutrition, and many, many more. But pretty much anything you could think of that could be found in biomedical papers. And yeah, this basically relationship comprehension and arrow assignment is done across all of these domains. Just a quick note about first stage of understanding relationship type before we sort of aggregate it into arrows between concepts. We classify our relationships using this pyramid, basically, right? At the very bottom, by the strength of causal evidence. At the bottom of the pyramid, there's a co-occurrence. We don't extract co-occurrence in this pipeline. We start with observations, and you could see different examples of how the strength of expression also increases. Just a quick note, there is, like I mentioned, the power to detect hypothetical statements and negated statements. And this is something that is not very obvious in its value. And it's in general, there's of course a positivity bias in literature, right? If you if you carry experiments and you don't find relationships or correlations for that matter, you would very rarely publish them. And to our analysis, there's no more than 10% of negated statements in publications. Vast majority is positive findings. But this, on the very famous example, of course, of chloroquine, is a powerful tool as well to put contradictory statements next to each other and develop your own opinion once you have this ability for refuting statements in this example. So I mentioned that we create a knowledge graph at the end of our text processing. What exactly our knowledge graph resembles, I'd like to go over in a few slides. There are different knowledge graphs. There is, you could start with knowledge graphs that express similarity, knowledge graphs that express descriptive relationships. These are often is a or also parent of narrower, broader sort of relationships expressed for various concepts. More information, I think, also could be found from similarity knowledge graphs and co-occurrence knowledge graphs, where various concepts are analyzed for co-occurring at the sentence level, paragraph level, or document level, and then statistically analyzed for how possibly strong the relationship, or does it exist also between them. And the fourth type is the one that we have. It's a directed computable knowledge graph, which we create from the relationships that we extract. And this type of knowledge graphs contains a very close to human level understanding of language, knowledge, which is derived from close to human level understanding of language. Of course, to create this knowledge graph, our relationship extraction is critical to it. And the powerful side of it is, of course, being able to do that on free text, on unstructured, semi-structured, and also structured data sources, and bring them together. And once you have this power of integrated data, the knowledge graph really grows powerful. And this is like, again, another basic view on the few nodes and relationships in there. Having this knowledge graph now allows us to really make a difference, because by querying this knowledge graph, there is a wide spectrum of questions that our users can ask. The questions which they can't ask in any other tool today, really. The fact is like I mentioned, is in the fact that we have now more than 240 million relationships which are domain agnostic in the knowledge graph, which is directed and computable. And these are a few questions that few examples of what sort of questions we can answer. Immediately, what are the biomarkers for X? What is the prevalence of X? What are the research trends for various concepts? What is the landscape for disease X treatment, and many more. It is natural, of course, to have a network structure, because these questions in a way are inherently network analysis questions. And on top of the few examples that we saw in another slide, very powerful thing is, of course, to be able to run network analysis here too. And we do this throughout in our product. We enable our customers to ask common cause, common effect questions. For example, what are the common treatments of multiple diseases? What are the common side effects of multiple drugs? And they could see them as Venn diagrams for intersections, overlaps, or unique evidence for a specific concept. Few notes on the ontology that we use, because this is, of course, central in knowledge organization. You could extract all entities you want, but if you cannot recognize them well and then place them in a structured ontology, vocabulary, or dictionary, there is very little integration power that you would have. For that reason, we are using the Unified Medical Language System. It's a umbrella ontology which integrates today more than 200 specialist vocabularies, including SNOMED, NESH, MEDRA, and many more. This is a tremendous work done by the National Library of Medicine in the US. I think for over 40 years they have been doing it. And they really allow multiple domains of science, biomedicine, to basically talk to each other through this structured knowledge. This is the end of my slide deck. Before I would go to, I should have finished the presentation in full with this, but since I already spoiled the slide, I just wanted to say that there are great news we are very proud to share in the company, and we're growing right now very rapidly. If you or somebody you know are get excited about what we're doing, please let us know. And let me, in the meantime, go to our platform. I think, can I quickly ask somebody to confirm that we are in my Chrome browser?
Yes, yes, I can see your browser.
Thank you, George. So this is the landing page of Causaly. And on top in the navigation bar, the first thing you see is various search options and different modules. What we will look at is simple and advanced search, but there are other things, like you can see PICO stands for patient intervention comparator outcome. It's a clinical domain of research, and then there's epidemiology domain, which studies prevalence and incidence of various diseases. What this reflects basically is, apart from wanting to transform how people work with evidence, with literature, we want to also build sort of an operating system for biomedical research, where people would be able to come to our platform and really ask a very wide spectrum of questions, not only for entity-to-entity relationship, causal relationship questions, but also other clinical real-world evidence, health economics questions. But let me continue. So in the center of the page, there is a search box that we will use now, and below there are just a few quick links for our users if they want to see the latest evidence regarding coronavirus. Let me run a simple search. Let me actually ask for causes of lung adenocarcinoma. It's the one I used in the slide example. It's the one I also rather often use. I am now at the results page. Just a quick note here, of course, apart from causes and effects of, there are other things that I could have just quickly defined for my search. Before going into the results page, I want to also mention the UMLS ontology sort of aspect that we incorporate. If I click on this show more for concepts, you could see different concept names that were incorporated in this search. What we basically are doing, we're leveraging parent-child knowledge from ontologies that we have to increase the recall of people's search, right? So if they search for lung adenocarcinoma, we also make sure that they various types, stages of disease progression are included, and the various adenoid cystic carcinoma of lung concepts are also there. But if you are not interested in specific concepts here, you also have the power to basically not look for them. But let's look at the results page. And as I mentioned, this is basically a bird's eye view on the full landscape, actually, of things that affect lung adenocarcinoma. We do say causes there. Not all of these are causes from a very formal hard causality definition, right? These are not measured causes of adenocarcinoma. These are all the concepts that affect and as well are causal to the disease. There are slightly more, 6,000 concepts, because I did not ask for biomarkers as I showed in the slides, but I rather ask broader. So if I scroll, you can see categories here, pathologic functions, neoplastic processes, findings, molecular dysfunctions, and various concepts which belong to these categories. And I keep scrolling. I can keep scrolling and seeing many other things. For example, organic chemicals, biologically active substances. These would be sort of treatments or also drugs which might, let's say, lead to a specific disease. And if I'm interested in something, I can click here on gefitinib, for example, and I could see evidence immediately from where we have extracted this information. This paper, for example, its evidence is in the title itself. The paper is about efficacy of gefitinib for Indonesian patients with lung adenocarcinoma. There is evidence from conclusion discussion section here, for example. Therefore, gefitinib treatment for patients with lung adenocarcinoma resulted in a marked survival benefit. That's why our system basically assigns a downregulate relationship here, and many more. On the left-hand side, sorry, actually, let me finish about the dendrogram. The size of the circle here represents how much evidence we extract for specific relationship. And the CT icon here means that there is at least one clinical trial where a specific chemical is being studied in relation to the disease. On the left-hand side is something that I could use to narrow down my search, to scope my search to something more specific, because 6,000 is still something that is rather hard to navigate. Of course, I can say I want gene or genome in be included, amino acid sequences, then also here, enzymes, receptors. And this actually would basically be my biomarker search, right? I'm just saying I want to look at these specific things. And what I end up with is with biomarkers of lung adenocarcinoma. And more, you can filter by data sources, you can filter by article sections. So we have PubMed, Medline, clinical trials in this out-of-the-box Causaly version, and others. Let me quickly show also something regarding evidence sorting. I'm sorting here by article count. I can sort by evidence score as well. This would basically incorporate the amount of evidence we extracted for relationship and would factor in linguistic strength of these statements that we have extracted. And you could see that the order of circle size doesn't comply here anymore with this way of sorting. If I click on the temporal analysis, I would be able to see how these various evidence emerged over time. In this case, I could extend, but in this case, these are the latest several months. What the size of the circle again represents is how much evidence we've extracted, and the placement of the circle indicates when we first discovered this relationship. And if I look for something very latest here, from April this year, this would be the latest findings, basically. So let me click on metal 1 gene. There's only one evidence that we have extracted precisely from the sentence. These are various papers that do not have direct relationship between these concepts, but still mention them together somehow. And if you look at the sentence, it says, taken together, these results suggest that METTL1 may be a potential diagnostic marker for LUADs, lung adenocarcinoma in this case. And this sentence is coming from conclusion section. I can look another one, MYG1 gene, and it says MYG1 may promote the proliferation, migration, invasion, as well as inhibit the autophagy of LUAD cells. So these are, of course, very relevant, and in Causaly, we have not encountered such evidence before when we were machine reading these tens of millions of documents. So we present them as new findings, most recent findings. This is example of the simple search. Let me go and show a few examples. Okay, Zoom is interfering. Let me admit this person. I'm not sure if I was authorized to do so. Let me do a few searches in the advanced search. Advanced search allows you not only to ask for concept-to-concept relationship, but also scope your search deeper. For the sake of the example, let me ask for causes of prostate carcinoma. So everything again that affects prostate carcinoma more specifically. For that matter, I'm leaving this box empty, and I can just, actually can just click search. And what we would see is very similar to what we saw in the simple search, right? There's 17,000 actually concepts here related as effectors to prostate carcinoma. And what I can do now is here, I can scope further. I can say, I want to only see studies which have used immunohistochemistry as a technique, immunohistochemistry. So from 17,000, we went down to 3,000 concepts, but let me actually go to article tab, which shows, which combines findings from direct relationships that we saw in the dendrogram, and also for when concepts are found together in various fields in the paper. So there are a lot of still results, but if I click here, I could see that these effects of prostate cancer, carcinoma, are studied in papers which performed immunohistochemistry on the tissue samples. So, and you can do more here, right? You can define country, you can define age groups, and other diseases, comorbidities, different treatments, and so on. Advanced search would also be a place where we would ask more complex questions, like the one I actually mentioned. Let me try that one. Ibuprofen, aspirin, and let's say, what is a paracetamol, which is acetaminophen. And let me define here upregulate. So the example questions in the slides I was given is, what are the common side effects of these three drugs? It might take a second to find them. So we are in the article tab still. However, if I go into relationships tab, I would see a delta analysis module here. So if I click on it, I would be presented with a Venn diagram where A, B, and C are the three drugs that I have queried. If I'm hovering over this part, these are side effects that are for side effects, I need to define disorders first. This is everything. So this question basically says, which disorders does these drugs upregulate, right? And if I'm hovering over B, these are unique side effects of aspirin. This is intersection of aspirin and ibuprofen, and in the middle intersection of all three. So I can click on them and I could see immediately 373 various disorders which were reported for all three of the drugs, and hepatotoxicity is, of course, the biggest of them. But you can see inside that paracetamol, by the number of evidence, there's a lot of evidence showing how paracetamol leads to hepatotoxicity, right? And paracetamol indeed is known for its very toxic effect on liver. Unlike, same for ibuprofen, for example, there's little evidence. It could lead, of course, however, it's not a common side effect of ibuprofen. And same for aspirin. If I, though, look at something like gastric ulcers, here, of course, the suspect changes, and it's aspirin that is well known to in the way how it can damage our stomach digestive system and cause gastric ulcers. And just to maybe add one more, I can go to Multihope. This is also a network analysis question. And let me just use one of the template queries that we have here. What I can ask in Multihope, because I'm querying basically a network, I can ask what are the causes of a specific concept. I can also have different modes here of how relationships go. But for the first example, what I'm asking here, how could obesity lead to cancer through this category of concepts? And these are various hormones, right? And I'm aggregated by the middle column, and I could see that there is relationship between obesity and leptin, for example, and then there is a unidirectional relationship between leptin and malignant neoplasm of breast cancer. So this activity is inherently a hypothesis generation, and could also be a mechanism of action understanding, which is quite relevant, of course, for research. There's, of course, a lot more and more that I can show, but I think I should stop there. There might be quite a few questions, and I would be very happy to address them.
Thank you, Artur. Thank you. It was very nice showing the demo as well. I think it helps understanding what you're doing.
Thank you.
I always have a lot of questions, but I prefer to give the floor to other people. Otherwise, I will just monopolize the questions. So any questions from anybody?
Thank you very much for the presentation. I have actually two questions. The one has to do with the relations you're using. Probably you mentioned it, but I missed it. But for the concept, you said you're using UMLS in order to map all the concepts in a specific format. For the relations, do you have a predefined list, or how you identify that two relations have the same meaning, etc.? So
yeah, that's a great question. It's I didn't go in detail about it. I just mentioned on high level this pyramid, but indeed that's what we do. Yes, when we extract relationships, we immediately try to understand what sort of a relationship it is, because it's one of the major building blocks in then defining the ultimate arrow assignment between the concepts, right? So when we extract the relationship, we classify it into one of these. There's more, right? This for the sake of the example, I showed these 5, but there are multiple classes into which relationship is immediately classified before we propagate our information extraction.
Yes, it partly answer it. So you categorize, you put its relation to one category, right? If I can understand. So the main types of relations are, part of them are if it's causal, affectors, etc. association. Is that right? If I understand correctly.
Yes,
okay.
That's correct. And these, the way we assign it's developed in-house. It's we didn't use some. We didn't use some standard way. Let me say that. And these are further used as one of the factors to define the arrow, right? So it's the linguistic, lexical aspect of the relationship, then the biomedical context that we get from UMLS, and then bringing them all together, we decide which arrow to assign for the relationship in the knowledge graph.
Okay, thank you. And then I have a different question, if I may, George. Yes. Okay. So this has to do with the evaluation. So you mentioned that you, first of all, if you have how you evaluate how good the extracted knowledge it is, you mentioned that there's a way to take some feedback from the users, right? But other than that, have you done any evaluation study to see how good is the network that you build and release?
Yeah, that's a very important question. That's the question we very frequently face, of course, when talking to customers, especially new customers, right? They always ask, how do you measure yourself? How can you say that you are good enough for our use? And that's a very valid, very important question. So from a very technical standpoint, going back to this natural language pipeline, there are stages where the accuracy is lost, the information could be lost. And for each of those, of course, we develop gold standards and baselines, and we continuously improve our language models, right? There are both symbolic and machine learning aspects to this pipeline. And whenever we do a change, there's immediately an understanding of what diff it's causing there, right? So there's a quality assurance stage and measurement stage. However, if I can measure how good my relationship extraction works, and then if I measure how good my entity linking works, and then my error resolution, it still does not tell you how good the end evidence is, right? And I can give you an example. For example, for error resolution, our precision on the mixed set, on basically agnostic of categories, is 89 to 90%. For chemicals to diseases, for example, our precision is 97%. However, still, this number doesn't speak for how well we did some other things, right? So for this end-to-end testing, what we do is we compare ourselves to human performance, because there are a lot of targeted reviews. There are systematic literature reviews that people do, and every time there is such work, it's a project in a company, right? The project starts. People go scope, screen documents, and then after a few stages, they start extracting evidence. And we try to measure ourselves to those results, and customers would have to see for themselves if we are good enough. And depending on the study, of course, we shown different results over time as well. We used to be worse than people. More often, we are getting better and better at this over time. There are things that still humans would find and we wouldn't find. However, our powers, of course, is in machine reading 30 million documents, right? So there would always be things that we find and humans don't find. So it's in both domains, which also shows that humans are not, they don't define gold standard, right? And the combination of the two could really make a lot of difference.
Okay, great, thank you. And just to clarify, for the individual modules that you evaluate, you have created manually some data sets, or you're using state-of-the-art data sets to evaluate each of the sub-modules of your pipeline?
A bit of both. Predominantly, it's our in-house data sets, which have context and representations that are important for what we're building. But there are some things for which we use existing benchmark and data sets are there. For example, for abbreviation extraction, right? There's no need for us to create something specific. We just use the state of the art and compare ourselves to these baselines.
Okay, thank you very much.
Thank you. Thank you.
Okay, then maybe I will have the chance to ask some of my questions.
Please, George, yes.
The main question that I have, and I think it applies more generally to those technologies, is has to do with the current trend of deep learning. How much of what you presented today can be done or supported by deep learning technology, and how much of it requires manual rule descriptions or even learning of rules or anything?
That's a very good and actual question that we in the team often discuss. I think in theory, of course, everything could be done using deep learning, given that there are enough of time iterations, configuring also the models. However, on the practical side, we unfortunately cannot just define the problem that we want to do, semantic information extraction, and say there is one model where we can just throw all this and it will learn and it will deliver. And the problem is very pragmatic, right? It's the performance is not good enough for end-to-end purpose achievement. There are definitely fields where deep learning already performs state-of-the-art in document classification, largely in entity recognition, and in those, of course, there is no point to be seeking anything else. However, for example, relationship extraction, it's very important that we have transparency and full control over what we do. And deep learning is unfortunately not very strong in both of those, right, compared to symbolic or rule-based systems. So when our users see a sentence and they say, you have not extracted it correctly, we would immediately know why. And if our users would want to see for regulation purposes, how was this, is this process deterministic? Can you explain the process of information extraction? We would also say yes. Those are a few aspects. And then the other one is that we could develop a symbolic system, which for this relationship extraction outperforms any other deep learning models that we tried, right? And try to also develop for that matter. So combining these factors, we have a symbolic system where we also know the canvas of the rule set, and we were able to build it also scalably in the way where we can see when will we reach human single sentence causality reading and comprehension performance. Given certain team and time, I could tell, for example, when we could achieve that. With deep learning, my control levels would not be the same, right? If I had something that doesn't work, I would need to think about my parameters. I would need to think about my training sets. I would need to think how to change things, and my response cycle would be completely different. So for that, I'm being more pragmatic about deep learning. However, other things like, for example, arrow resolution, once there is a large body of data which is of high fidelity, which we actually have right today, I believe there could be models trained that could assign, for example, arrows, given all the factors that you propagate into these models. So I'm rambling here a bit, but coming back to your question, George, I think there are quite large amount of things. I can't say exactly how many, what portion, but large amount of things that could be and are done with deep learning. But there are some things that practically for our work, I don't think it would be a good decision for us to go with them. If that answers your question. In the near future.
Thank you. You mentioned, you basically implied trust to the model, and you said how explainability is an important aspect in trusting the decision or the recommendation of the system. And you also discussed earlier with Natasha the issue of evaluation and performance of individual models. That's upon provenance, being able to see where this comes from. Yeah. So there are many different sources. I mean, the fact that you have a pipeline of, as we can see in this slide, a pipeline of different things may add up to difficulty in explaining, also to accumulation of error. So there are many different sources of uncertainty, if I may put a label to this. Yeah. How do you handle that in a situation where you eventually are judged by the outcome by what the user is looking for.
Yeah, I agree. Every stage we add, especially if it's a potential accuracy loss or information loss stage, we create a new degree of freedom, right? And by Occam's razor, and the least you can create, the least you have to have, that's the number you should have. And creating more increases the complexity and then this cascading effect of error propagation and many other things. However, when driven by necessity and not done for the sake of structure, where stages, they carry a specific semantic mission, and they are modular, which is a great advantage because when you build your pipeline in a plug and play scenario, where you can like take any stage and dissect and understand it very well, then again, it, to be honest, only increases your control, right? For any problem coming from the end of the pipeline, I can find out, I can tell where that problem is happening. I can take that specific module and I can interrogate it and understand what went wrong and what shall I do about it. So this modular plug and play approach actually helps us more rather than creates problems today. And the complexities are more for us on the practical side are automating it and making it faster, because having so many stages and then machine reading 30 million documents, you can imagine it's it takes a bit of time. So these are more operational aspects that are troublesome. However, on the questions that you mentioned, yeah, it actually makes a difference, more positive difference for us rather than negative.
It is related to the previous one. How do you handle contradicting evidence? So if you have different sources telling you opposite things, how do you present that to your users?
It's a very good question. Let me maybe quickly show this. Actually, COVID-19, for example, this hydroxychloroquine example I can show. So when we extract evidence, our job is to translate this evidence to the user as with the full transparency as much as possible. We do not make decisions on what to show, for example, or not. We can only make decisions on how to sort, and that could affect what users see first, right? But what we extract is everything is available, right? And we present this through these arrows. So for hydroxychloroquine coronavirus COVID-19 example, you can see how many various evidence we extracted. There is a lot of evidence saying that it downregulates COVID-19, so you could consider it as a treatment. However, there's also examples that a lot of evidence which refutes that statement, and others showing that they upregulate relationships, sort of. So if I wanted to see refuting statements next to each other, I could do this, and I can just look at them. So here they say efficacy of hydroxychloroquine, it's in title, but here in the conclusion it says HCQ post-exposure prophylaxis did not prevent SARS-CoV-2 infection, and many others. So this is basically the way we present this evidence.
Okay, thank you. Thank you. I mean, I could go forever. Each answer has, gives 10 more questions, but I think that just means that what you're doing is something very, very interesting, at least for me. I'm sure for many other people as well. So I would like to stop here and thank you again. Thanks for everything, and wish all the best to Causaly and the expansion that you're doing at the moment. I'm really glad that that this work is being is reaching a maturity level that you're reaching. And thanks very much for the presentation again.
Oh, thank you, George, a lot for inviting. It's very valuable for us. Makes us very happy to know this work is also interesting for you, that we have a channel of communicating and also maybe working together. Yeah, thanks a million, Artur Saudabayev, and Any questions, George, you might be still having, I'm very happy to discuss separately.
Yeah, I'm sure we'll have the chance to discuss again.
Yep.
Okay.
Thank you all. Thanks for attending.
Thanks, everyone. Bye-bye.